Okay, so meiosis versus mitosis. Meiosis is sexual, genetic information is mixed, and meiosis can produce sex cells. Um, Mitosis occurs because the cell can only get so big. If the volume can't be satisfied by its surface area of absorbing nutrients, it must split somewhere. The phases of meiosis are prophase. The chromosomes condense, becoming shorter and thicker. The centrioles assemble, and spindle fibers uh, attach to the centromeres of the chromosomes. The nuclear membrane starts to dissolve. Metaphase. Chromosomes line up at the equatorial plate. The nuclear membrane completely dissolves. Anaphase. The centromeres divide and the resulting chromosomes, formerly called chromatids, move to opposite poles of the cell. An identical set of chromosomes moves to each pole. Telophase or telophase. The chromosomes lengthen again and the spindle fibers dissolve and a nuclear membrane forms around the chromosomes. Okay. And then cytokinesis is when the plasma breaks apart. Meiosis are cells that have a haploid number of chromosomes are called gametites. Meiosis produces gametites. Diploid cells have a normal amount of chromosomes. Um, Homologous chromosome is a pair of chromosomes with similar shape and gene information. A zygote is the formation of a male and female sex cell. A tetrad is a pair of homologous chromosomes. A synapsis is the formation of a tetrad. Crossing over is the exchanging of genetic material. Okay, and there are two types of meiosis, so we'll start with one and then go to two. So prophase 1, synapsis and crossing over happens. So the replicated chromosomes condense, homologous chromosomes come together through synapsis, and crossing over occurs. Chromosomes are attached to the spindle fibers. Then there's metaphase 1, which is like the chromosomes line up at the equatorial plate, the homologous chromosomes. And then there's anaphase 1, which is the chromosome separates from its homologue and moves to the poles, to the opposite pole. And then there's telophase 1, which is the nucleus is completely divided, or the nucleus completes its division. The chromosomes are still composed of sister chromatids, and the cytoplasm divides after telophase. Then there's prophase 2, so the centrioles move. So the centrioles in the two new cells move to opposite poles, and new spindle fibers start to form. The chromosomes become attached to the spindle. Then there's metaphase 2. The chromosomes line up at the equatorial plate. Anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. And finally, telophase 2, where the cytoplasm separates, leaving 4 haploid daughter cells. The chromosome number has been reduced by half. These cells may become gametites. There's asexual reproduction. Sexual reproduction, formation of gametites, gametogenesis. Spermatogenesis starts at a spermocyte. It divides twice to produce 4 sperm. Oogenesis starts with an oocyte. It divides once and creates a good egg and polar body. And then it divides again and creates a good egg and 3 polar bodies. Non-disjunction and errors. A trisomy is having 47 chromosomes. A monosomy is having 45 chromosomes. Down syndrome is an extra chromosome 21. Turner syndrome is a single X chromosome. And Klinefelter syndrome is 2 X's and a Y chromosome. Cancer cells are called with uncontrolled are cells with uncontrolled and unregulated growth. They tend to ignore commands from the body, and metastasis, like a cancer cell of a tumor, is moving elsewhere. Genetic crosses and problems. There's genotype versus phenotype. So Punnett square, monohybrid, dihybrid, sex length, multiple alleles. ABO types, transfusion, universal donor and recipient, sex chromosomes, which is on page 153. After your time. So yeah, 153 is good for the checking out the Punnett squares. Given ratio of genotype slash phenotype, DNA structure, lethal dominant goes away fast, lethal recessive retains in pot for a while. And that's it for this video. I'm going to let it run a couple seconds so I don't have to repeat myself.